As all the downstream analysis will be performed next on the Seurat object, so we will load the Seurat object that was created and saved in our previous session. So now we will load the Seurat object pbmc. So we will use the read RDS function in R to load the Seurat object pbmc.rds and run. The Seurat object has been loaded. Now we will add the gene annotation to the Seurat object. So add gene annotation. But before we add the gene annotation to the Seurat object, first of all we will check the annotation slot within the Seurat object. So pbmc assays, and then we will access the peaks, and within the peaks we will access the annotation slot and run. Now look at the output, null. This means that no annotation has been added. So now we will add the gene annotation to the Seurat object. But a question is, why we add the gene annotations to the Seurat object? The answer is very simple. We add the gene annotation to the Seurat object because single-cell ATAC-seq data largely contains the chromatin peaks. It doesn't tell us about where the promoters and transcription start sites and exons are located within the genome. So by adding the gene annotation to the Seurat object, we can easily locate all these regions within the genome. First of all, we will extract the gene information from the ensemble based database. So extract gene information. So we will use annotation and get ranges. The get ranges from ensemble database is a convenient Signac function that pulls the gene information from the ensemble based database. So now ensemble database, and we will use the version 86, and then we will load. Now this line of codes pulling the gene information from the database. It will take some time. So now all the gene information has been extracted. As we retrieved the data from the 10x genomics that uses the UCSC chromosome naming system, so we will change this to the UCSC chromosomes naming system. So convert chromosome names to style, so we will use the given line of code. Chromosome annotation and run. This will fix the naming system of chromosome according to the UCSC style. Now we will take the gene annotation with the correct genome build. So take the gene annotation, genome, and run. Finally, we will add the gene annotation and to the Seurat object. So add gene annotation and to the Seurat object. So annotation PBMC, which is a Seurat object, and run. The gene annotation has been added into the Seurat object. Now finally we will check that whether the annotation has been added or not. So PBMC assays, picks, and we will access the annotation slot. So as you can see the output and the console, first of all this code return a null output, but now you can see the annotation has been added to the Seurat object.